Welcome to Spatial Threads from PredxBio, a conversation on the groundbreaking developments in spatial biology with some of the pioneers in the space. In this series, we delve into the very exciting area of spatial biology, where researchers are diving into the intricate complexities of cancer and other diseases, leveraging the tools of spatial proteomics and transcriptomics. We will hear from the leaders in the space how they're advancing the understanding of the hidden circuitries of advanced cancer and other complex diseases, making a difference in treatment and patient outcomes. Today, we have Dr. Arutha Kulasinghe from the University of Queensland kicking off our Spatial Thread series. Dr. Kulasinghe has pioneered spatial biology for the last decade or so, and he has actually published many world-class studies, including ones in lung cancer and head and neck cancer. He has been a champion in the areas of spatial transcriptomics and proteomics. It's a delight to be here today with Dr. Arutha Kulasinghe. Welcome.
Thank you, Dusty, for the kind introduction and the invitation to the first iteration of Spatial Threads. I'm super excited by it. I'm joining you from sunny Brisbane, Australia, and welcoming your guests to this really great forum I think you've put together.
Terrific, and thank you for joining at this early hour in Australia. So very honored to be with you. So Arutha, I've got to know you over the last couple of years quite well, and beyond knowing you as a collaborator in spatial biology, I can also call you, one of my friends, which I'm really honored to do. And I know you've been in the space of spatial biology now for a while. You're one of the leaders in this space for the last 5 years. But before that, just kind of getting into your personal journey a little bit, you were in the area of liquid biopsies. And tell me a little bit about that journey and how you made the transition onto spatial biology and the synergies between the two.
Absolutely, Dusty. So I started my journey about 10 years ago, did my PhD in the liquid biopsy space. And so we were looking at predictive biomarkers, looking at the blood, circulating tumor cells, circulating tumor DNA in the blood of advanced stage lung cancer, head and neck patients, and trying to identify, the cells in those samples that were prognostic or predictive of therapeutic benefit. We learned really early on that whilst the blood gives you a snapshot, really trying to understand what's going on fundamentally, we need to go back into the tumor and look at the tumor microenvironment. And so for every one of our blood-based studies, we tried to pair it where we had matched patient tissues and blood. And so that's what sort of initiated the work. And then I, about 2017, 18, got into the spatial transcriptomics, initially proteomics space, where we were looking at, 20, 30 proteins at a time, pseudo bulking, not really single cell. And when we got the data back of these, we call them high-plex assays during that period, once we saw the data and we could see, the degree of infiltration, the amount of information we were gleaning from this, that sort of set the precedence for, this is where we really need to focus on, because that's where immune cells fundamentally function, right? So there was a beautiful paper a couple of years ago, and it showed that immune cells function in the tissues, and so really understanding the microenvironment is probably going to be a better predictor that you can then match with your blood and other peripheral samples. And so we started off with this low plex sort of 20, 30 proteins, and now we're doing hundreds of proteins, whole transcriptome, single cell, in advanced stage lung cancer, head and neck, a bit of work in skin cancer, and we're doing some studies in breast and ovarian cancer too. During COVID, obviously, we wanted to contribute to the global efforts, and so working with collaborators in the US and Brazil, we've been able to look at the lungs, the hearts, the brains, and the placentas of patients, and really understanding the differences in COVID and flu and the multiple pandemics, how it affects adults and children and so on. So we've got a number of infectious disease studies, and the major focus of the lab is obviously cancer.
I had a similar journey in the space of liquid biopsies before I came into spatial biology as well. And, one of the things that's really been remarkable is this ecosystem that you have been able to build at the University of Queensland with the Spatial Biology Center, which is heavily funded, by the Australian government. Tell us a little bit about what it took to build that ecosystem over there.
Yeah, so part of the challenge in understanding these complex biological data sets is we're looking at the histology from pathology specimens, and we're trying to overlay the molecular information over that, right? And so, 10 years ago, we could get away with a bulk approach or a single cell approach, and your endpoint is a t-SNE or a UMAP and so on. But now we really we really need to understand where this duality is, where we're putting this molecular information over your pathology and your histopathology specimens. And so where these cells are in space is really important. And so to interpret these data sets, you traditionally need a pathologist, multiple data scientists, computational folk, biostatisticians, immunologists to understand, what are these cell types? Are they real? Are they artifacts and so on? And so we've been building this team over the last 2 to 3 years where we've now got that really synergistic team of oncologists, pathologists, immunologists, computational biologists. So now we can collectively interrogate these diseases. And so you need that team. And so part of the problem was how do we build that team? And so we've built that out now, and we've been able to get philanthropic funding and Cure Cancer and national funding to support this. And we've set up the Queensland Spatial Biology Centre, which is purpose-built on a hospital site so we can run clinical samples to understand clinically focused research questions. And so I think it's that next iteration of moving these tools from sort of discovery biology through to translational and clinical research. Our previous director, Ian Fraser, developed the Gardasil vaccine. And so we're based at the Translational Research Institute and the Wesley Research Institute. And so we're very translationally focused on developing these tools to really take it from that bench to bedside.
Terrific. And you've seen the field of spatial biology really develop over the last 3 to 4 years, I would say. And the fact that both Nature Journal and then Nature Methods, carried very important articles on it. Nature actually had a full journal devoted to spatial biology last October. So, and I think you've really accomplished a lot, and you've basically epitomized that it takes a village to really make a difference and to do anything that has true purpose behind it. So, what do you think the area of spatial biology has accomplished so far? We saw a blog, I think, not too long ago from Eric Topol talking about spatial medicine and how the researchers in Max Planck actually were able to help a patient. So tell me a little bit about your perspective on what spatial biology truly has accomplished.
Yeah. So I think we're still in the early days, Dusty. These tools have, traditionally in the last, 5 years, we moved from this discovery biology, and now we're seeing the real impact come through with these translationally focused clinical studies. And so that beautiful study that Eric Topol discussed as the dawn of spatial medicine, it's a beautiful term, I think, that we can now use because it's actually being used in the clinic. And so I think the next few years, what will happen is we will see a lot of movement in these, low plex spatial scores, spatial signatures. If you look at the ASCO sort of clinical trial data, all the clinically linked multiplex IF signatures were 15 to 18 plex or below. And so, if we develop tumor stromal predictive biomarkers and they're sort of in that, 6 to 10 plex space, those can potentially move on to companion diagnostic assays. And we can see that there's roadmap for that. There's been an FDA-approved spatial signature for ovarian cancer. It's a predictor for a DNA damage drug from Macrovon Therapeutics. And so we can see some of these LDT, CDX assays, low-hanging fruit coming through. And I think the next wave will be on really focusing on clinical cohorts where we can look at retrospective or prospective studies of, patients that did well on drug versus patients that didn't do well on drug, teasing apart those predictive biomarkers and then building it into some form of CDX assay that we can turn around pretty quickly, right? So the experimentation plus the analysis needs to be in a clinically relevant time period. And I think that'll happen. But right now, I think the focus is on running the multi-center cohorts to build the robustness of those spatial scores spatial signatures, whatever term you want to call it. I think that's the next iteration, and that's what we'll see happen. We're already seeing pockets of this happening in the US and Europe and so on. But the real value of these signatures will be in the reproducibility across multiple labs, right? So a lab in Boston should be able to generate the same result as a lab in LA or a lab in Brisbane, for example. And so running those same samples at multiple sites, running the same workflows, generating the same data, and being able to then use that will be really building the evidence to adopt these. I think the other really exciting part is, the pathology community will start to get excited by these tools that are being developed, and I think it plays into hopefully the digital pathology space.
Terrific. And I think you have mentioned multimodal data, playing more and more in the spatial arena. So talk a little bit about that. How do you see the next 5 years evolving with the integration of multimodal data and that actually being used down the line in the clinics?
Yeah. So I mean, when you look at, those predictive scores, those spatial signatures, they ideally telling you, you know, these patients are responsive due to the expression of these proteins in the tumor, for example, positive across these 4 or 5 proteins, this patient's likely to respond. But we're not really gleaning insights into mechanisms of resistance, mechanisms of sensitivity. And so that's when we need to dial it back and do the discovery-based work on those same samples to then tease apart what is happening at a mechanism level in the patients that are resistant to drug, patients that are sensitive to drug. And the only way to do that is using a transcriptomic data. And so when you start to do that, you can start to now overlay why the patients are resistant to this drug, but also that may tell you what drugs the patient might be eligible for, right? So if we're identifying pathways that are druggable, in those same samples where we've got protein signatures, that becomes this multimodal data set. So I think in the study that was published in Nature by the Max Planck, you could see that they identified this JAK inhibitor that would have been important for this skin condition, and so they acted on that in a clinical trial setting. And so you could see how identifying the mechanisms in those and the pathways that are involved, because the pathways, whilst there might be novelty in identifying those pathways, there might be existing drugs that are actually make those patients druggable. And so, you might not only be giving anti-PD-1, PD-L1 patients those drugs, you might identify a combo therapy that might boost the effect of your immunotherapies and so on. And so I think that's how we sort of need to think about bringing in the transcriptomic data, the multi-modality data, to really define these resistant phenotypes and these sensitive phenotypes.
Sure. And from a standpoint of challenges in spatial biology, I think you mentioned standardization, right? The labs in Boston and Brisbane and Mumbai should be able to do these tests in a similar way and really have results that can be universally applied. Besides that, I think you just mentioned about the mechanism of action. Just generating data for data's sake does not really, work out for oncologists or patients. So how do we kind of get better at it? Would you say these are the most challenging issues that still remain to be resolved in spatial biology?
Yeah, I think the computational challenge is one of the biggest challenges we have today, and building, really building out spatial informatics companies, tools, analysis pipelines, platforms is going to enable, any wet lab researcher to be able to generate the data and be able to analyze the data on site. And so I think the spatial informatics field is developing. There are some really exciting companies, including PredxBio in the space. I think that are developing novel tools to understand the microenvironment in these tissues. But the next piece is how do we, run the same sample at multiple sites and have that analysis pipeline set up, or is it a cloud-based pipeline? Do we upload the data onto the cloud, and is it analyzed in a straightforward pipeline? And then we're all able to interpret that live or on the cloud with the pathologist, with the immunologist on the calls to really understand what's going on in this patient's tumor. And so we can see that in the pathology space, that's already happening, low plex assays, 3, 4, 5 plex assays. They've got reproducibility studies around the world. There was a study that was led by Janice Taub called the MIDA study. There are a number of other studies in this space that will come out this year, really standardizing multiplex immunofluorescence for the field, but then that'll come with the sort of caveat of spatial as a bigger tool. And I think there are multiple global efforts for data standardization, interpretation, and so on. And I think more consensus around this would be useful, but it also is a challenge because the technology companies are boosting their assays every 6 months, and so developing the tools to keep up at that pace has been challenging. But I think over the next year or two, what we'll see is standardization of what we call spatial assays to something similar to what we think of when we say genomics or whole exome. We think of a single assay or two assays. Once we have whole slide, single cell, whole transcriptome, and maybe high-plex proteomic data, that allows us to then standardize to one assay and then be able to compare across and run the same assay across the world. I think those efforts will start happening.
Absolutely. I think it's the integration of all of these data sets coming together that's going to really unravel the complex circuitries of cancer in the next few years. You've also been a global leader in the sense that your collaborations, a lot of them are very global, and you brought together a fantastic group of people in Brisbane just in December in a conference unlike anything that I've really seen before. So tell us a little bit about the conference, what it took to get all these leaders together, and also what came out of it.
Yeah, it was a really exciting meeting, Dusty. We had about 50 international leaders in the space of spatial biology, probably the largest concentration of spatial leaders in the world, all of them jumping onto 14 to 20-hour flights to come to Brisbane. What we learned at the meeting was fascinating. We had a coffee one of the mornings, and we had folk that were from, oncologists, pathologists, immunologists, sort of AI folk, just sitting around having a coffee, having a chat. And, they had different caps on from wherever they were working, right? But the fundamental question they were trying to answer was, how do we move these assays forward to the clinic? And so you could see that the barriers around, I work for this company, or I work for that technology, or this technology, that was left at the door. And they were collectively thinking about, how do we cohesively streamline this process so we don't spend the next, couple of years developing tools that aren't being used, but actually focusing on the endpoint, which is the patient and the clinic and so on. And so a lot of those questions naturally happened, which is great. A lot of collaborations were set up, which is really great. So I think we'll see the fruits of that meeting this year and hopefully in the years to come. Again, a lot of like-minded people that fundamentally understand that we're trying to build better tools for patients and understanding, why patients respond in certain ways to therapies. I think if you have patients at the center of your research, and I realized this when I spent some time at Peter Kuhn's lab at USC, is that they had patients in the labs doing research, right? So when you have that set up and you come to work and you're surrounded by, your colleagues, but also patients that understand that, I am trying to further understand my disease. And that really, left an imprint on me. And so what we've tried to build locally is a patient-focused research team where everything is focused around the patient. And so what are we trying to understand about their tumors, about therapies? And it's not about the patient's immediate journey, it's about the collective journey and what we can do for future lung cancer patients or so on. And so I think what happened by everyone coming for multiomics was that they sort of left their, caps and egos at the door, and they sort of collectively, everyone thought about the problem. I think there'll be some really, really exciting collaborations and results from that meeting.
Well, that's terrific. And that's exactly what we need to break down the barriers and the silos and see how we can all come together to really make a difference. And that's part of our mission here at PredxBio as well. So, hey, it's a privilege to have this conversation with you, Arutha. Really appreciate your time, and we'll be in touch. And thank you very much. So that was Dr. Arutha Kulasinghe at the University of Queensland.
Thank you, Dusty.